The oldest written texts are about 5,000 years old, and we are only knowledgeable about them because their Sumerian authors used clay tablets, which they kiln-baked into permanence. Similarly, long-lived texts were produced by the Etruscans, who hammered their best ideas into gold plates. The switch to papyrus and paper as a medium, while brilliant, made written words short-lived, and now the transition to electronic texts has reduced that fleeting time to a New York minute. What we write on our iPads is doomed to disappear fast, and baking these tablets in a kiln is certainly not going to help. Nature has the same problem. Its text is written in DNA, an extremely fragile medium, more like tissue paper than a clay tablet. Yet some of our genes have been used to dictate essentially the same protein for more than 1 billion years. How then do living cells ensure that their genetic instructions remain readable for future generations of cells and organisms? Nature's solution to the instability problem is to repair the DNA text. Because most cells have 2 copies of the text, an intact copy can be used to restore the corrupted version. But for this system to work, cells need an editor to monitor the DNA for any kind of change, paragraphs that are broken off, sentences that are interrupted, words that are changed. How do cells sense damage in their DNA, and who is this editor? The Lasker Basic Award is given to 2 exceptional scientists who discovered the answer to this question in the 2 main types of organisms in our biological universe. Evelyn Whitkin first found the solution used by bacteria, and decades later, Steve Elledge worked out how this is done by eukaryote cells like ours. In the 1940s, before the Lasker was started, Evelyn Whitkin was a young left-wing radical studying at NYU, where she demonstrated against NYU's policy of editing all black players out of its basketball team before a game in the apartheid South. In line with her politics, she turned down NYU for her graduate studies and selected a Russian geneticist at Columbia University, Theodosius Dobzhansky, as her thesis advisor. He worked on fruit flies, but Whitkin wanted to work on E. coli, which, as she had learned from a paper by Max Delbruck and Salvador Luria, could be used for genetics. Dobzhansky therefore suggested that she work for a while with Delbruck at Cold Spring Harbor Laboratory. Whitkin loved Cold Spring Harbor and stayed there for more than a decade before moving to SUNY Downstate University and then to Rutgers University. Delbruck gave Whitkin a UV lamp and an E. coli culture and told her, go make mutations. Not a carefully considered thesis project, but enough for Whitkin, who started her career with the same independence, courage, and grit that later helped her negotiate the patriarchal world of science. Although UV kills E. coli, Whitkin was able to isolate a few mutant strains that survived. These mutants helped her dissect the mechanism by which E. coli monitors its DNA. And this story is really an amazing detective story because in the 1950s and the 1960s, the E. coli DNA damage response field discovered a dazzling diversity of phenomena, including the induction of bacterial viruses, the induction of error-prone DNA replication, and changes in cell growth. And to most scientists, these phenomena were not obviously connected, but Whitkin, with phenomenal deductive insight, understood that there was a central principle lurking behind them and provided the data to back up her view. Like Hercule Poirot, who at the end of the detective story invariably gets everybody together in one room to baffle them with his analysis of the murder case, Whitkin organized a small conference where she, without Poirot's arrogance, presided over the final reveal moment of her field. That meeting established the concept of the bacterial SOS response, a term originating from the creative biologist Miroslav Radman. The central player in the SOS response is a protein called REC-A. REC-A binds DNA, but only when the DNA is not in its usual double helix form, a clear indication that the DNA text has been corrupted. Once REC-A, the editor, has found the corrupted text, it helps to inactivate gene repressors, and this releases many target genes to express their protein products. Thus, a wide array of proteins spring into action to orchestrate the repair of the DNA damage and ensure the stability of E. coli's genetic text. Years later, Steve Elledge started his scientific career actually working on the SOS response as a graduate student with Graham Walker at MIT. But Elledge became more interested in how eukaryotes sense the damage in their DNA, a question he pursued as a Howard Hughes investigator, first at Baylor and later at Harvard. Elledge, a virtuoso geneticist, worked out a way to isolate mutant yeast strains with altered DNA damage response, thus identifying, as did others, a set of DNA damage response genes. But what do these genes do? Most researchers expected something like the bacterial SOS response, but Elledge had a different idea. He hypothesized that eukaryotes, being more complex, use something more like a burglary alarm, with a detector that senses the DNA damage and then rapidly signals to remote control posts, who then alert the right DNA repair crews. To test this idea, he studied the proteins that are made by the DNA damage response genes, and his key discovery was that some are protein kinases, proteins that add phosphate groups to other proteins, which is a signaling mechanism by which cells can quickly activate whole sets of proteins. He found that a single alarm arising from a protein kinase at the site of DNA damage is sent to another protein kinase by way of phosphorylation, and the second kinase then changes the whole cell's physiology so that the DNA is repaired before the cell continues in its division cycle. Elledge also discovered how this eukaryotic system senses DNA damage. Once again, finding that nature's editor recognizes DNA that is not in the double helix form as a signal that something is wrong with the text. Many of the DNA damage response genes studied by Elledge are now getting great attention because they prevent cancer, as is the case for BRCA1, while others protect against aging and neurological disorders. The discoveries of Witkin and Elledge opened up new fields and are found in every biological textbook. But better than being in textbooks, which, being electronic, are passing bits, their discoveries are written in stone. They form the foundation of our understanding of how DNA is kept stable for billions of years. Perhaps the next time we fix a corrupted computer file with a backup copy, we will note that it was nature's principle of sensing and repairing a mistake that got us to this point. Please join me in congratulating these wonderful scientists, Evelyn Witkin and Steve Elledge.